what I was telling is, we are using visible light to see the using the light microscope now. So visible light usually ranges from about 400 nanometers to 700 nanometers, right? So can we change that? No. So usually the maximum resolution power is half of these 200 nanometers, right? So we can't do anything about. I also don't know why half that they have. I don't know what physics is. So maybe on that wavelength, so maybe half I don't know what it is, right? Okay. Right. So whatever it is, we can't change this uh, visible light rate, so that we can't increase the resolution power or magnification in the light microscope more than this, right? So it's a kind of a fixed value. So this is a limitation, right? I write the answer there for the question. It's asking what are the limitations to increase the resolution power of the light microscope, right there. Resolution power of the light microscope. Resolution power of the light microscope. Is limited by the wavelength of light. Right there. Next question is asking, right, add one more sentence to that same thing. Resolution power is inversely proportional to, resolution power is inversely proportional to the wavelength of light. Write this equation there. Resolution power Right? Right. Now we'll give you a little time to study those that we discussed now. Take 5 minutes and study quickly. Remember the definitions. Those will be question in your question papers. Are you all done? Let's go to the next one. Mm, we are now going to talk about the electron microscopes. You know what? This is not easy to handle. This is a big equipment which takes more space, and you need some expertise knowledge to use this one. Okay. So usually in a compound light microscope, you will be using the glass slides to observe. No? So here we have to use pre-prepared slides to observe the specimens in the electron microscope. You have to treat them prior with some heavy metals, and specially prepared thin slices of specimen will be used to observe. But in light microscope, if you want, you can now also make a slide and observe, no? Isn't it? But it is not easy like that in the case of electron microscope. So here in the compound light microscope, we use glass lenses to focus the light beam. But in the electron microscope, we use magnets to direct the electron beams. Their electron beam is used. Okay. So since we had some limitations in light microscope, like we can't increase the resolution power or the magnification furthermore due to this wavelength of light, we are now going for another option, that is electron microscope, which is later developed than the light microscope, right? So there are two types of electron microscope we will be learning here. Number one is transmission electron microscope. We call it as TEM. And here number two, scanning electron microscope. That is called SEM. Scanning electron microscope. So these two having two different mechanisms of generating the picture. I'll first take transmission electron microscope. There is a question asking two types, no? Write that answer there before I erase. Hi, I'm erasing the last one here. Transmission electron microscope. Let's see what's happening there. An electron beam is sent towards the specimen. So here we are keeping the specimen, right? So specimen is here, right? So these electrons are passing through this specimen, but not everything is going out. Some of these electrons are coming out, right? So there is a detector on this side. This detector is detecting these electrons. What is the amount of electrons passing through? You know what? If the specimen, some places there may be thick places, so that less amount of electrons will be passing to the other side. Some places will be thin, so that more electrons will be passing to the other side. So detector is taking this information and sending it to a computer. In the computer only this image is created according to the information sent through this detector. So that the computer is generating a picture which is a two-dimensional picture, a black and white magnified image from this TEM, transmission electron microscope. Can you all come to the cover page? Go to the cover page. There Look at the bottom two pictures. What do you guess? Which is TEM picture? Yeah, bottom right hand corner. That is the TEM right there. Electron micrograph obtained from TEM. We call electron micrograph as the picture that is obtained from the electron microscope. Usually in the some Peradeniya University they have one, as I know. I sometimes maybe in those slim pick those things they will be having. I'm also having no idea where they are having. Our university we haven't had one. Okay, right? Colombo no. I'm from Colombo no. Right? Okay. Only very few. Peradeniya I heard because they need some maintenance with liquid nitrogen and all that. I think not everyone is doing that one. Some knowledge is needed, right? Okay. Hari. So this is giving a black and white image, but in the books that you may have seen, colored electron micrographs. How do you get that? Here you are using a computer, no? Using computer softwares, you can put any colors, right? And the initial picture getting from this is a black and white picture. If you want, you can use computer softwares to use any of the colors, right? Okay, any color you can put, no matter, right? If you know, let's say chloroplast, if you know it is blue, green color, you can use green color. But if you want, you can put blue even. Yeah, that's what I told. The initial picture is black and white. There's no color generated here. The computer is the one which is putting color. If you want, you can put any of the colors. Okay. Alright. Then scanning electron microscope. Scanning electron microscope, SEM is the short term. You are using that as well. So here what happens is an electron beam is sent to a specimen. So here is the specimen, let's say. It is scattering these electrons which are coming to this specimen. Some of the electrons are absorbed by this, but most will be reflected by this. So detector would be somewhere here that is detecting the electrons which are scattered from the surface of the object. So that surface is not even. Some places will be scattering more, some places will be scattering less. So that according to that, this will be creating a image using a computer. So it is also kind of a black and white magnified image, but different from this one is that is giving surface details in 3D manner. Here it's a 2D picture. Look at the first page again. Yeah, the bottom left hand corner one, that is taken from uh, scanning electron microscope. So that is a scanning electron micrograph. Alright. Now go to the page that I have given two pictures of these electron microscopes. Yeah, the mechanism. Look at that one. Yeah, those two. Check whether you can understand that too. You don't have to draw anything, okay? For your understanding, only those are given to you. What happened to your friend, Putte? May that girl is not your friend, eh? You came alone. That girl who is coming time to time online as well as physical. What is her name? Anne, eh? What happened to her? No, long time no see. Now everyone come and read the electron microscope section completely. Alright, now you can start writing the answers from electron microscopes. Write on your own. Can අපුතේ heavy metals because the electrons are used to observe this one, no? So that electrons have something with metals, no? So that's something related with that. Okay, otherwise they are not going through it, no? Hurry, quickly write the answer so that we can go to the next section. Are you guys usually free on Thursday evening? Usually, are you free on Thursday evening? You? No, to have an extra class. Not physically, online I can have in the evening. I'll let you know. I'm so sure. You have a class, ne? That's poor day, have I? Ah, practices. That's poor day. It's poor day. You do have practices on poor days? Check it in and what about you, Pute? Yours all, huh? What do you write for the nature of the electron micrograph obtained from a transmission electron microscope? A magnified image, black and white image, what else? Two-dimensional image. Sign up. You should have tell me no, Putte. So you have to come to the next class, ne? When did your father call me, Putte? I don't remember, ne? Did he come to the class and talk to me? I'm so sorry. I didn't know that from your which batch you are coming. You should have tell me early. When did he talk to me, Putte? I don't remember the incident exactly. Is it fine? Shall I give the next link also to you? It is at 3:30. When? When? I'm asking when. Did he talk to me or the class? Wait. Okay, maybe. Right. That's fine. You use the next link to join at 3:30, right? So we have the class. Right. Okay. Let's check the answers sooner. What is the briefly describe the mechanism of action of the TEM. A very thin specimen is used. The specimen is stained with heavy metals. Beam of electron is passed through the specimen and they are displayed on a screen through a detector. Have they given detector word? No, no, don't write don't change what is given in the book. I'll read the answer given in the book. Let me change it. Mm, transmission electron microscope. These things should be there. I read one of the students' answers here. Mm, it is used to study the internal structures of cells. A beam of electrons, you can start with that point as well. A beam of electrons is passed through thin, specially prepared slice of material. A very thin specimen is used. Specimen is stained with heavy metals which attach more to the certain cellular structures than other areas. Image reflects the pattern of electrons passed through the specimen displays on a screen. Okay. While the electrons pass through the specimen, more electrons may get displayed in the regions where the structures were densely stained. That part also should be there. Big paragraph is coming up. Alright, done. Next question. Next section, as I told, transmission electron micrograph. Next one, briefly describe the mechanism of action of electron scanning electron microscope. Right, you have to write there. In this instrument, a fine beam of electrons is reflected from the surface of the specimen. Specimen is mostly coated with gold prior to observation. That is also a heavy metal. Okay. Here the specimen scatters many electrons, whereas others are absorbed. This instrument is ideal to observe the surface wave of or the wave in the three-dimensional appearance. Okay, that part you wrote. Next one is asking the nature of the scanning electron micrograph. You had write magnified image, three-dimensional image. Image is a black and white image. Okay. Hi. Nature. Yeah, that one we can exclude. Yeah, true, true. Three-dimensional. If you have written that, that's fine. Don't cut, but you can exclude that one. Yeah, that's not mechanism. Yes. Hi. Next, asking the maximum magnification and resolution of electron microscope. Maximum magnification in is 5 into 10 to the power 5, and maximum resolution power 2 nanometers. Hi, write both answers there. Hi, let's go to the next one. What are the advantages of using electron microscope over light microscope? What do you think? High magnification, high resolution, more clarity of the image, isn't it? Next asking, what are the advantages of using light microscope over electron microscope? Actual color of the image can be observed. Movement of the object can be observed. Na? Actual color of the object can be observed. A movement of the object can be observed. What else? Easy to handle. No expertise, knowledge is needed. Sometimes we need, because light microscope, some light microscope we need to plug and put the light. Okay, don't try that. Yeah, no need of expertise, knowledge, low maintenance cost. Write the next answer alone. Next answer and go to the next table given regarding the comparison of light microscope and electron microscope. Read it carefully and then copy it to the table, right? Okay. Right. Let's go to the next section. We have now about cells and cellular organization. No? So, you know, early days they didn't have much more information about cells now. So, they developed certain microscopes and observed cells now. So, initially, Robert Hooke observed the cells. Robert Hooke. Robert Hooke observed the cork cells. They had some structure like this. He said it looked like a beehive. These are dead cells that he observed. Okay. Hi. Then, later on, a compound light microscope was developed. Anton van Leeuwen Hooke. That one is the one who observed about microorganisms and recorded first, right? There may be certain other people at the same time who have observed, but they didn't record anything about them. He is the one who actually observed and recorded about microorganisms. And then thereafter, later on, they developed a theory called cell theory. There was few scientists involved in it. You learn about cell theory in all of us, right? Did you? Hi. So what are the things there in the cell theory? All cells are made up of one or more cells. All organisms, sorry. All organisms are made up of one or more cells. ඊළඟට? Yes, very good. Yeah, the basic structural and functional unit of life is the cell. Also, he said, also another person said that cells are arising from pre-existing cells. Let's see which of scientists involved in this finding. Come to the text here. Right. It says the scientist Matthias Leiden, he studied about plant tissues and concluded that all plants are made up of cells. And also Theodor Schwann, zoologist, concluded that all animal cells are made up of cells. So altogether, this statement is created, all organisms are made up of one or more cells. And then Rudolf Virchow said that cells are arising from pre-existing cells by cell division, and then who said that the basic structural and functional unit of life is cell. Altogether, according to these statements, they could conclude that the basic structural and functional unit of life is cell, right? So, who are the scientists involved here? Matthias Leiden, Theodor Schwann and Rudolf Virchow, right? What did Matthias Leiden did? He said he studied about plant tissues and he concluded that all plants are made up of cells. Then Theodor Schwann, all animals are made up of cells. And then Rudolf Virchow, all cells arise from pre-existing cells. Okay, right. Let's go to the questions from that section. I said everything now about those things. Yeah. read that section complete before writing answers. That paragraph in page number 24. Did you ask anything, Shaman? Yeah, yeah. Yes, no, no, no need, no need. No one would ask this. Right. Sometimes I would have switched off the mic. I don't remember that I switched off the mic before I teach the previous thing. So I'll be repeating what I discussed quickly. So I told about the finding of the microscope result, like allowed to see more structures, the internal structures of the cell, and to study about the cells. And firstly, Robert Hooke observed cork cells. He examined cork cells and he saw that some beehive-like structure there. So that is he named as a cell, those single component, right? So that is actually, these are dead cells now. So thereafter, Anton van Leeuwenhoek developed the compound light microscope. He is the one who is first to observe and record the details of microorganisms. What kind of microorganisms he observed? Euglena, paramecium like small organisms, no, unicellular organisms. And thereafter, cell theory was developed by three scientists. What are the three scientists? Matthias Leiden, Theodor Schwann, and Rudolf Virchow. What is the finding of Leiden? All plants are made up of cells. Then Schwann, all animals are made up of cells. And then Virchow, yeah, Virchow, cells are arising from pre-existing cells. Now we are okay with that part now? Alright. Now we have the common features of prokaryotes and eukaryotes. Now, is it so? See, cellular organizations. Yeah. So here in this section we are learning there are two types of cellular organizations. We call them as prokaryotic cellular organization and eukaryotic cellular organization. Prokaryotic means there's no organized nucleus. And eukaryotic means have an organized nucleus. And you may have seen bacteria. They are belong to prokaryotes. Bacteria, cyanobacteria and archaea belong to prokaryotes. They do have chromosomes in the cytoplasm itself. This is a circular DNA. So there's no nucleus. Usually our chromosomes are located at the nucleus. So here they don't have a nucleus, but this region which is present, the DNA is present, we call it as nucleoid. Alright? Nucleoid region, right? So there are so many differences between these two cellular organizations, right? But still there are similarities too. What about viruses? Are they belong to any of the cellular organization? No, they are not belong to any of these cellular organization, right? Hi, let's go to the similar features of all these cell types, the prokaryotes and eukaryotes. It says these cells are bounded by plasma membrane, which is a selective barrier. So that plasma membrane is a common feature for both prokaryotes and eukaryotes. Do you all agree? Right? Their structure also similar. They are sharing similar structure of the plasma membrane, and they have within the cell they have semi-fluid jelly-like substance which is called cytosol. Subcellular components are suspended within the cytosol. Again, what is inner to the cell membrane, we call them as subcellular components. So inside the liquid component, the subcellular components are present, right? Okay, the liquid component we call it as cytosol. They carry DNA as genetic materials. Both of them have. Prokaryotes also have, eukaryotes also have. What about viruses? They do have genetic material, DNA or RNA, any one of these. They are not considered as living because they are not showing all characteristics that we discussed in the first lesson. Okay. Uh, oh wait, right? Archaea prokaryotes, right? Ribosomes are found in all cells. Small ribosomes, severous ribosomes are found in both types of cellular organization. Understood? Harry. Now read and study them and quickly copy them into the question answer. Right. Now we are coming to discuss the differences between prokaryotes and eukaryotes. There's a lot of differences between these two types of cellular organization. Right. So here, uh, organisms belong to these categories: bacteria and archaebacteria. Cyanobacteria also belong to bacteria category. Eukaryotes, who are the examples? Plants, fungi, animals, protists. Correct? Those are belong to organisms which are having an organized nucleus. So they say that, talk about form. Form means unicellular or multicellular. So these guys are mainly unicellular. These ones are multi and unicellular, both are there. Many are multicellular. Right? So fungi such as yeast is unicellular. Some protists, amoeba, euglena, paramecium, diatoms are unicellular. Right? No. These not protozoans, protists. These are protists, broad category. This is having ulva, sargassum like big ones also, macroscopic ones also there. So some protists only unicellular. Yeah. Amoeba, protozoan. Unicellular ones, protozoans. Yeah. Okay. Right. So evolutionary origin. Who evolved first? Hmm. So this evolved before, you know that, 3.5 billion years ago. And these guys evolved 1.8 billion years ago from prokaryotes. From them only they have evolved, right? Then cell division. Remember, since they don't have nucleus, they can't do mitosis or meiosis. These guys are having a nucleus, so that they will be dividing by mitosis and meiosis. So instead of mitosis and meiosis, these guys are showing simple cell division such as binary fission. This one show mitosis and meiosis. Got it? That part okay? After that, then about genetic material. This part is important. These guys are having circular DNA. I'm writing like a comparison. Circular DNA is present. These ones are having linear DNA. These guys, DNA is not associated with histone proteins. Histone proteins only found in eukaryotes. Okay. Then what else? Found in the nucleoid region. This one's found in the nucleus. Got it? I'll take a little time to study that one. That is important. Remember very well, this is not mentioning about the proteins association of prokaryotic DNA. In the 7th unit, they are clearly saying that prokaryotic DNA are also associated with proteins other than histone proteins, right? Right? Usually we say a chromosome is DNA plus proteins. Right? If this protein is histone proteins, we call them as true chromosomes. True chromosomes are found in these guys. These guys don't have true chromosomes, right? But proteins are there. Okay. Histone proteins only found in eukaryotes. Got it? Right. Take a little time and study. Yeah, having binary fission. Fission is there in, no, sorry, wait, fission is not there. Fragmentation, budding, parthenogenesis like asexual reproduction methods are found in eukaryotes, not fission. Fission is not. Okay. All right, all done. I'm going to the next one. I'm erasing this part. If you want anything more, that which I write in the board, I'm writing extras now. So if you want anything, just note it. Okay. I'm not telling one by one because I have given the table to you. All right, let's go to the next one. The types of ribosomes. I told that ribosomes are common to both, but 70s ribosomes, small ribosomes only present in prokaryotes. Eukaryotes, we have both 70s and 80s both. Right? Okay. And then organelles. Right. Understand what I am telling to you guys. Organelles is the big category. No, sorry. Subcellular components is a big category, which is inside the cell membrane. All the structures found, we call it as subcellular components. Understood? Among that, there are subcellular components which are bounded by one or two membranes. Are bounded by one or two membranes. So these we call them as organelles. Are so for example, mitochondria, ER. Likewise, you can write any examples which are bounded by membrane. So there are subcellular components which are not bounded by membrane. Example, ribosomes we can take. Alright. So here it says no membrane bound organelles present. Agree? Consist of few subcellular components. Example, ribosome. Right? So the other side, membrane bound organelles and other subcellular components are present. Great diversity of organelles. Different types of organelles are present in them. Internal membrane craces present and usually associated with respiration, photosynthesis and nitrogen fixation. It's something like this. These guys are having cell wall. Inner to the cell wall, they're having cell membrane. Cell membrane is having some infoldings. These infoldings, they do have enzymes to do photosynthesis, pigments to do photosynthesis, enzymes to do photosynthesis, as well as respiration. They having uh, enzymes. So if it is doing enzymes, we call them as mesosomes. Enzymes are present inside this. But if you know that these eukaryotes, they use mitochondria for what? For cellular respiration. Then they use chloroplast for what? Photosynthesis. And these guys don't have chloroplast or mitochondria. Got it? That's what I explain here. Cell walls, peptidoglycan is present in bacteria. Bacteria cell wall component, peptidoglycan. Then archaea having proteins and that is polysaccharide. But they are telling it as a different thing, so we have to remember as it is, right? Proteins and polysaccharides. Correct? Right. Then here, that part, okay. So this guys who is having cell walls, plants are having what is fungi is having, and also protists also having cell walls, right? All the information are not given in the table. You can include if you want. Plants are having cellulose. What else? Hemicellulose, pectin. Fungi have chitin. Protists are having cellulose, silica, alginic acid. Sorry? They were organisms prior. They were prokaryotic organisms engulfed by a big cell, and then they were living together with a big cell in a eukaryote. Alright? That's how it happened. There is a theory called endosymbiont theory that you are not learning. It says that mitochondria and chloroplasts were separate organisms before unicellular. They will be engulfed. They have been engulfed by a big cell, and now they are living together mutually in that cell. That's why Prokaryotes have 70s ribosome. Inside mitochondria and chloroplast, we have 70s ribosomes. And also, both have DNA, circular DNA are there. And also they have a double membrane. So when they are engulfing, that another part of this, the host cell also coming now, right? Okay, understood? Right. Next one, it says about their flagella. So here, their flagella, let's say this is a prokaryote, their flagella is like this. They are not enclosed by plasma membrane. Therefore, we call it as extracellular. Understood? These guys are, let's say, sperm cell. Sperm cell, there is a big flagellum like this. So it's covered by plasma membrane, right? So we call them as intra within the cell now. Intracellular. Understood? Intracellular. Got it? Then these not having microtubule arrangement. No microtubule arrangement. Are it? But here there is a microtubule arrangement. 9 plus 2 structure. You will learn in coming up lesson. 9 plus 2 structure. That means there are 9 triplets and 2 microtubules in the middle. Hmm. Are it? So regarding size also there's a difference. So this is smaller, about 20 nanometers. Yes. This is about how much? What's 200 nanometers? Are it? So bigger. Understood now? Both are there to locomote. That function is same. The next one, it's talking about the respiration, mostly done by internal membrane foldings, and mitochondria are therefore anaerobic respiration in eukaryotes. Photosynthesis, no chloroplast, takes place in membranes which no stacking. That means grana are there in chloroplast. No, there's no stacking of membranes here. Chloroplasts contain membranes which are usually stocked into lamellae or grana. Nitrogen fixation, remember very well, prokaryotes are the ones only having ability to fix atmospheric nitrogen biologically. So eukaryotes can never fix atmospheric nitrogen. Are they? Okay. Why are they fixing atmospheric nitrogen, children? Yeah, in the nitrogen cycle, the plants cannot take as nitrogen N2, so that it has to be converted into ammonium or nitrite in order to absorb by the plant, so that nitrogen fixing bacteria or cyanobacteria are converting them into uh, absorbable, like soluble forms, right? Okay. Right, now we will take a little time to study this one. This table is extremely important, I would say. Every time many questions are coming from this section. So in the book, tables you have to by heart, okay? So try to study now, and then next week also we'll give time to study. I have given questions from that table, no? Isn't it? Have I given? No, 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 no. In the table, check. Haven't have given, no? I'll continue the questions now. It will take time to study the table, but it's worth by heart in the table. Go home and rewrite and practice. Okay, this is always question. Which question? Some part is not printed there. How many pages are missing? 3 pages. Mm. You also took the new one, eh? You don't have the other tubes, eh? Go down and bring another set of tubes and come. You have to bring your given ones, okay? Yes, but you? Like, naked DNA means DNA not associated with histone proteins. අන්තිම ටික කිව්වොත් අන්තිම දෙක දීලා තියෙනවා. කවුරුත් නැහැ ඔන්ලයින් එක අහගන්න. අර ළමයා 26. ඔව්. හරි. Next one. Do prokaryotes do photosynthesis? Yes. Cyanobacteria, some cyanobacteria, some membrane infoldings / mesosomes. Mesosomes. Eukaryotes, Dahane. What is the question? Prokaryotes, yeah. If online student miss anything because mic is turned off, I didn't see it. If you want anything, just leave me a message, okay? What is the type of structures which involve in photosynthesis in prokaryotes? Membranes which show no stacking, or else you can say membrane infolding is also correct. Do yeah, internal membranes. No, not internal membranes. Membranes which what is it? No, just write what I am telling right there. Membranes which show no stacking or / membrane infoldings, right? Do prokaryotes have the ability to fix nitrogen? Yes. Do eukaryotes have ability to fix nitrogen? No. What is the cellular organization of cyanobacteria? Prokaryote. Alright, I'll finish at this point. We'll meet on next day to study about the cellular structures, plasmamembrane and everything, right? So there's a lot to study about those things, and bring the tubes. I need the glue and a scissor to cut and paste. So, and writing books also needed. If you want, you can maintain the papers as well, that's fine. But bring to cut and paste those pictures. I have given pictures to you now. Hi. Okay. So everyone, don't forget to bring those. Next, we'll meet on next Saturday.